Hello everyone, let us talk about the procedure for the next experiment entitled Acids and Bases. For the materials, we will need the following. 1 normal hydrochloric acid, 5 ml to be placed in a test tube. Then 1 normal acetic acid, 5 ml again, 1 normal ammonium hydroxide, another 5 ml, and 1 normal sodium hydroxide, 5 ml. So we will need 4 test tubes for this. Reagents. Red and blue litmus paper will be provided in your box. Drops of methyl orange, so this is around 1 ml. These are indicators, methyl orange and phenolphthalein indicators, which you will place in 50 ml beakers. Take note, you need only 1 ml. Okay. Pieces of aluminum shavings will be placed in your box. And for the ferric chloride and chromic sulfate, you will need 3 ml to be placed in the test tube, and a 10 ml of 6 normal sodium hydroxide in another test tube. So this is the procedure which we are going to modify. Initially, it calls for 5 ml of water and 5 drops of dilute HCl. Shake and cautiously taste. So we will do away with tasting. Dili na tamo taste. Okay. Instead of 5 ml water, we will be using a smaller amount because we will make use of this apparatus. This is called the spot plate. A spot plate, also called a reaction plate, is a laboratory tool made either from ceramics or plastic. Each plate consists of many cavity-like depressions in which only a small amount of reactants can be added at a time. So, instead of 5 ml of water, we will be using 5 drops of distilled water in each of the well. Okay, so 5 drops of distilled water first into 3 wells. Add 1 drop of dilute HCl to each of the 3 wells containing distilled water. Test the solution in the first well with a small piece of red and blue litmus paper. The second well with a drop of methyl orange indicator. And the third well with a drop of phenolphthalein indicator. You will do the same with the rest of the wells. Yung next set of 3 wells will be used to test the acetic acid solution. So again, the same procedure. Lagyan ng tig 5 drops of distilled water. But this time, sa second set of wells, you use 1 drop of acetic acid solution. Tapos ganun pa rin, litmus paper, methyl orange, and phenolphthalein indicator. Sa next set of wells, 5 drops of distilled water again, but this time, use 1 drop of sodium hydroxide. Okay, carry the test again with litmus paper, methyl orange, and phenolphthalein. And the last wells, we use ammonium hydroxide, 1 drop of ammonium hydroxide, and then the same set of acid-base indicators. Okay, so this is it. Repeat the procedure using acetic acid, sodium hydroxide, and ammonium hydroxide. Then fill the table with the observations you have made in the above procedure. Okay, so this is the table. Again, we strike out the tasting part. So in each of the cell, you will place the color reaction with the various indicators. So this is part 3 of the exercise. You will need 2 test tubes, one to contain 5 ml of hydrochloric acid, the other to contain 5 ml of acetic acid. And to each test tube, we add a pinch of aluminum shavings. Okay. It says do not cover and observe what happens. Okay, you are asked in which test tube is there more rapid reaction. You are asked to write the equation showing the product of reaction. Take note, this is a reaction of acid with metal. This is part 4. Again, another 2 test tubes. But this time, you will place ferric chloride in one test tube and chromic sulfate in another test tube. Now, we modify this so that instead of 5 ml, we use only 3 ml. 3 ml. Now, to each test tube, add 1 normal sodium hydroxide until you obtain a precipitate. Okay. Take note of the color of the precipitate. Okay. Take note of the color of the precipitate, and you are asked to write the equation. This is the instrument that I'd like to introduce to facilitate separation of the supernatant from the precipitate. This is called the centrifuge. Okay, so instead of this portion, withdrawing the supernatant liquid by using a dropper, we do not do this anymore. Instead, we make use of the centrifuge. Centrifuge and decant the supernatant into an appropriate container. To each precipitate left in the test tube, add 5 ml of 6 normal sodium hydroxide and shake. Okay, so diba meron tayong test tube that contains 10 ml of sodium hydroxide. Paghahatiin nyo lang yun so that each test tube containing precipitate will get approximately 5 ml of 6 normal sodium hydroxide. Okay, and take note what happens to the precipitate, write the equation, and ngayon pa lang, research what is amphoteric hydroxide. Okay, so this ends the pre-lab discussion.